Assalamu alaikum to everyone. We are going to start a new topic, chemistry of amino acids and proteins. You might be knowing that proteins constitute about more than 50% of the body dry weight. Proteins, the fabric of protein consists of amino acids. Amino acids, they are united with each other by peptide bond, which is a covalent bond. So different amino acids, they unite with each other by peptide bonds, and they ultimately form the dipeptide, tripeptide, tetrapeptide, pentapeptide, or the different polypeptides or proteins. Amino acids are the building blocks of proteins. Here you can see the structure of amino acid. Amino acids are a group of organic compounds containing two functional groups, amino and carboxylic. It is because of the presence of these two functional groups, amino and carboxylic. Carboxylic is an acidic group. So due to the presence of these two functional groups, we call them amino acid. Amino represents the amino group and acid represents the carboxylic group. So amino acids. The carbon atom, which is attached with the two functional groups, carboxylic and amino group, this carbon is called alpha carbon. This alpha carbon is attached with beside the two functional groups, is attached with the hydrogen and this R group. R group varies in different amino acids, and it is because of the presence of different R groups in the amino acids we categorize the different types of the amino acids, the different set of amino acids, the properties of amino acids, everything that is dependent on this R group. The amino group is NH2 basic, and the carboxylic group is acidic. Amino acids are joined by peptide bonds. So this is how the two amino acids are joined with each other. Here this is the peptide bond, where amino group of amino acid and carboxylic group of another amino acids, they combined covalently with the release of water, and they form the peptide bond. In proteins, amino acids are joined covalently by peptide bonds, which are amide linkages between alpha carboxylic group of one amino acid and alpha amino group of another. You can see here the example valine and alanine. Both these amino acids, when they join, carboxylic group of valine combines with the amino group of alanine with the release of water. When the water is released, this is the covalent bond, and here you can see the peptide bond will be valyl alanine. This is a peptide bond where both the functional groups are incorporated into the peptide bond. This amino group of one amino acid and another one is carboxylic group of another amino acid. Standard amino acids. All the proteins, they are composed of different 20 amino acids. We call these amino acids as standard amino acids because they have the genetic coding to be incorporated into the proteins. Those amino acids, there are around 300 amino acids in nature exist, but all of these amino acids are not standard amino acids. Standard amino acids are only those amino acids which have the DNA coding for the protein synthesis. So there are 20 standard amino acids. This is because of the universal nature of the genetic code available for the incorporation of only 20 amino acids when the proteins are synthesized in the cell. Proteins, the most abundant, 50% of the cellular dry weight. Proteins form the fundamental basis of structure and functions of the life. Proteins, as I told you, proteins are composed of the different amino acids. So the number of amino acids and the sequence of amino acids which is present in the particular protein, that defines the nature of the protein, that confirms the property, the structure of proteins. Proteins on complete hydrolysis with the concentrated HCl for several hours yield L-alpha amino acids. This is the common property of all the proteins. Therefore, proteins are the polymers of L-alpha amino acids. Functions of proteins. We categorize the functions into two groups: static, structural, or dynamic or functional. Regarding static functions or structural functions, everybody knows these include collagen and elastin found in bone, matrix, vascular system, and other organs, and alpha keratin present in the epidermal tissue. So this is a static function of the proteins. Regarding functional proteins, the functions of the proteins more diversified. Enzymes, all enzymes are proteins, and some hormones, proteinous hormones like growth hormones like insulin, blood clotting factors, immunoglobulins, our immunity, membrane receptors, storage proteins, and they have the genetic role. Proteins have the genetic role, muscle contraction, respiration. So these are all the functional properties of the proteins, and this is the structural properties of the protein. General structure of amino acids. The amino acids are termed as alpha amino acids if both the carboxylic and amino groups are attached to the same carbon atom. This is called alpha amino acid. Side chain represents by R, which is different for each of the 20 amino acids found in proteins. So this is only the presence of R group which differentiate between the different types of the amino acids, their properties. Otherwise, every amino acid will be having the same structure: alpha carbon and carboxylic group, amino group, and one hydrogen. Mostly the amino acids, they are present in our body in ionized form. Optical isomerisms of amino acids. If any atom, carbon atom, is attached with the 4 different groups, it is called asymmetric and therefore exhibits optical isomerism. All the amino acids except glycine, which is the shortest amino acids, having 2 carbon only, where R is equal to H, have optical isomerism. So every amino acid except glycine will be having optical isomerism. You can see here, this is asymmetric carbon. It is attached with 4 different groups. It is attached with CHO, with H, with CH2OH, with OH. So this is asymmetric carbon atom. So optical isomerism will be present in all those amino acids which are having the asymmetric carbon atom. And because the glycine is not having asymmetric carbon atom, so optical isomerism is not be exhibited by the glycine. The points which I underline. I usually must focus on these points. These are important regarding your MCQs in your prof. The structures of L and D amino acids. We classify amino acids as L amino acids or D amino acids according to the configuration of amino group. When amino group on any amino acid is on the right side, it is called D amino acid. And on the other hand, when amino group on any amino acid is on the left side, it is called L amino acids. In our body, most of the amino acids are L amino acids. In contrast to the carbohydrates, mostly the different monosaccharides in our body, they are D type. So D glucose, D fructose, D galactose, they are commonly present. So D category of carbohydrates is present in our body, and L type of amino acids, they are predominantly present in our body. Now we classify the amino acids. Amino acids are classified on the different bases. Amino acids classified on the basis of the structure, can be classified on the basis of polarity, on the basis of the charge they are having, on the basis of nutritional demand, whether they are essential, non-essential or semi-essential. They can be categorized on the basis of whether they are ketogenic, glucogenic, or having the both properties. So 20 amino acids found in proteins are divided into 7 distinct groups. The very first one is the amino acids with aliphatic side chains. These are 5 amino acids: glycine, alanine, valine, leucine, and isoleucine. When the R group is straight chain or branch chain, this is common to all amino acids. This portion, this is alpha carbon, which is attached with the carboxylic group and amino group and one H. This is H, this is glycine, the smallest amino acids. The R group is represented with H. So this alpha carbon will be having two same groups, one H and another one this H. So this is not asymmetric carbon. That is why glycine do not exhibit optical isomerism. So I want to say that this group is common to all the amino acids, every amino acid, 20 amino acids. This is the pink color is representing the R group. So different R groups are present on the amino acids, and they totally change the properties, the structure of amino acids. So when the methyl group is a straight chain or branch chain, these are called aliphatic amino acids. Glycine is a straight chain, this is a branch chain, this is branch chain. So amino acids with R chain consisting of the continuation of the methyl groups is called the aliphatic amino acids. Now the amino acids containing hydroxyl group, these are 3 serine, threonine and tyrosine. You can see here again, you can say this portion is common to all the amino acid. This is R group, this is representing R group. Now here on the R group, this is hydroxyl group. This is hydroxyl group. So the functional group of this amino acid is hydroxyl. In the same way, threonine has hydroxyl group, hydroxyl. Tyrosine is having hydroxyl group. We'll discuss in another slide. Tyrosine is formed just by the addition of hydroxyl group to phenylalanine. So phenylalanine having hydroxyl group is tyrosine. So all are having hydroxyl functional group. Although amino acids are represented either 3-letter abbreviation or single letter abbreviation, like this. Glycine is represented by GLY or with G. Similarly, ALA is for alanine or it is represented by A. Similarly, every amino acid can be represented by 3 letter or by 1 letter abbreviation.